Okay, so this is the second of 4 macromolecules screencasts that you need to watch. Now we're going to look at lipids. So lipids are primarily carbon and hydrogen. They do have a few oxygen atoms in them. Because they're primarily hydrogen and carbon, and if you think back to our intro to the chemistry unit, we know that those are hydrophobic molecules. They don't dissolve in water. So their roles: longer-term energy storage, insulation, lubrication, cell membranes, and hormones. We'll do some calculations to kind of actually establish this, but lipids are lighter energy than carbohydrates. So they're used for energy storage in organisms that move, because if we had to store all of our energy as starch or as glycogen, we'd be too heavy to get anywhere. Lipids are also used in insulation of the body, lubrication of tissues and joints, cell membranes, and hormones. They're essential to the body. So even if we cut out lipids from our diet, which we shouldn't do, your body would make them anyways, right? So we tend to, in North America, in our diet, sort of demonize one particular food group at any given time and cut it out of our diet. And then we end up, our body just has to make it for us anyways. So for example, you can eat a low cholesterol diet. Doesn't mean you're going to have no cholesterol in your blood, because your blood needs cholesterol for your cell membranes and for other functions. So the monomers and subunits of lipids, they don't really have any, because even though they are big molecules, they're not polymers. And there are 4 main types of lipids. We're going to focus on these guys at various points in the semester. So triacylglycerols or triglycerides are used for energy storage. These will be important next unit when we start talking about metabolism. Phospholipids, we'll talk about this unit when we look at membrane structure. So they are what's responsible for comprising the bulk of cell membranes. Oh, sorry. Sterols are 4 carbon-based rings attached to one another. And we looked at cholesterol when we were looking at functional groups. Different substituents on the 4 carbon sterol rings give each different sterol its properties. So estrogen, progesterone, testosterone, cholesterol, they're all examples of sterols. And then waxes have quite diverse chemical structures, but the thing that defines them is they tend to have super long carbon chains, and they're all solids at room temperatures. So these triglycerides, some of them might be solid at room temperature, some might be kind of soft solids at room temperature, and some of them might be liquid at room temperature, but all waxes are solid at room temperature. And they're made by cells to coat surfaces and prevent water loss and offer some physical protection. So most plants have a waxy cuticle of varying thicknesses on the surface of leaves in particular. So triglycerides are where we're going to spend most of our time. They're composed of a glycerol spine and 3 fatty acids. So this is my glycerol spine. Glycerol is a 3-carbon triol. So it's 3 carbons, and each of those carbons has an alcohol group on them. And then a fatty acid is an extremely long carbon chain with a carboxylic acid on the end. So the fat is the long carbon chain, and the acid is that organic acid part. What happens when they form is that an alcohol on the glycerol and the carboxylic acid react. They release a molecule of water, and they form that triacylglycerol. Similarly, we can break them apart. So we can take our triacylglycerol and break it apart by adding water across that bond. And the fats in your triglyceride can either be saturated or unsaturated. So most of you probably have heard at some point the term a saturated fat or an unsaturated fat. What that's determined by is how many hydrogens are attached to the carbons in the fatty acids. A saturated fat means that every carbon in the fatty acid that makes it up have the maximum number of hydrogen atoms bonded to it. And what that means is that the fat takes on a very regular zigzaggy shape. And because all three fatty acids in the fat are saturated for something to be considered a saturated fat, the fatty acid chains stack quite easily together, and in turn, neighboring fat molecules, neighboring triacylglycerols, stack quite neatly together. And that's bad for humans in large quantities because those can stack quite neatly at room temperature and clog your arteries. So I'll illustrate that on the board tomorrow when we go over this. Unsaturated fats have one or more double bonds in the chain. And what the presence of a double bond does is it introduces a kink into the chain. And that kink prevents the individual chains in the triacylglycerol from stacking neatly, but also neighboring triacylglycerols from stacking neatly. So they're kinky and they don't play nice with others. So these aren't as bad in terms of the fats that we can eat. And in fact, in some cases, they're good for us. So omega-6 fatty acids in particular are supposed to be good for our heart. Here's the illustration. I thought I had one in this note. So the saturated fats, in reality, they actually take on a zigzag pattern rather than this straight thing. And I'll show you that. And you'll do a lab to practice with some model building. So the saturated fats stack quite neatly because they've got this regular shape. Whereas here, when we introduce this double bond, it's just an area of stress on the molecule that introduces a kink, and these don't stack neatly. Phospholipids are very, very similar to triacylglycerols. Here's my glycerol molecule, and here's two fatty acids. But in this case, one of the fatty acids has been replaced with a phosphate molecule. And typically it's a phosphate molecule and it has some other charged group up at the top. So choline is pretty common, and we would call a phosphate or phospholipid that had a choline on it phosphatidylcholine. This means that the head of the fatty acid has this charged choline group, and the phosphate group itself is charged. So it has a polar end here, and yet these big, long fatty acid chains are uncharged and unpolar. So that gives the molecule a really unique property where it has polar regions and non-polar regions. And when you dissolve those in water, they line themselves up so the polar heads are facing out and can interact with the water, and the non-polar tails are facing in. So toward the end of our evolution unit in the phylogeny unit, we said one of the requirements for life to kind of originate on Earth was the existence of these protocells that are membrane bound. So the earliest protocells might have just been a micelle. Current cells line up so that the phospholipids in the cell membrane don't just form this single layer in a micelle, they form a bilayer. This is also how detergents work. So detergents in your soap have an uncharged area and a charged area. So the uncharged area picks up the grease or the dirt or whatever is on your clothes inside of it and dissolves in water with these polar heads. So sterols, as I mentioned, are all derived from cholesterol. They've got these 4 carbohydrate rings, 3 6-carbon and 1 5-carbon ring. They are involved in the cell membrane and in cell signaling, metabolism, and hormones. So the general structure is this, and you can see a few modifications and additions give you some completely different hormones with the same basis. In triacylglycerols and phosphates, ester linkages are what's used to connect the fatty acid to the glycerol backbone.